to the cloud. Okay, now let's share the screen here with this Histology lecture 2 of 3. Okay, so we are officially recording here. I do not like what I'm seeing on my toolbar. Okay, it indicates I'm recording. I don't believe it. Anyway, so we're done with epithelial tissues. Let's get to connective tissues now. So connective tissues, as the name implies, they connect things. They hold stuff together. They're the most abundant tissue type in the human body. We see stratified squamous epithelium on the outside everywhere, but underneath that, all the way down to the bone, you're going to have a lot of connective tissues. Different types of connective tissues. Bone itself may be considered a connective tissue by a lot of authorities as well. Okay, so with connective tissue, the cells are not going to be tightly packed together forming rows. You're not going to see a lumen nearby in all cases. They tend to be few and far between, just a few cells separated by wide expanses of collagen, which forms most of the extracellular matrix in the human body. So this stuff forms the physical three-dimensional shape of organs. Okay, so again, connective tissue, mostly extracellular matrix or ECM proteins. And whether it's a bone, a muscle, heart, lungs, liver, stomach, whatever, lymph nodes, hair follicles, this connective tissue is what gives organs their physical three-dimensional shape. So there's two major categories of connective tissue. And then within each of these major categories, connective tissue proper and supporting tissue, there's subtypes. So connective tissue proper, 3 types, you could argue 4. These are what we call fascia. They form what we call fascia. Fascia forms a cushioning boundary that supports organs, anchors the organs into the rest of the body, keeps them from going where they're not supposed to be physically. Supporting tissues are associated with bones and articulations or joints, and supporting connective tissues include bone, also something called cartilage. Okay, so most of these tissues is going to be one specific chemical compound, and that is a protein that we call collagen. So after water, which is 66% of our bodies by weight, collagen is the next most abundant chemical in the human body. The functional part of collagen, we have these 3 helically braided subunits, kind of like braided strands, braided cable. So we say it's a protein that's composed of multiple subunits, 3 of them. They braid together and they form what's called a quaternary structure to that protein. Okay, so an actual collagen fiber, it's not a little thing. They're huge. We can see them with a microscope. You can't see molecules with a light microscope. Even most organelles are too small, but sometimes we can actually see a collagen fiber. They're that big, that thick, that dense. Okay, so connective tissue proper itself broken down into 2 main groups, loose connective tissue types and dense connective tissue types. They're categorized on basically how dense is the weave of collagen fibers. Is it open and loose like cashmere or a cardigan knit sweater? Or is it really tightly woven like silk or a really high quality cotton broadcloth? So the 3 types of loose connective tissue are, number 1, areolar or loose connective tissue, which forms fascia, layer of connective tissue between organs, between muscle and muscle, between muscle and bone, between other organs. And then we have something called reticular connective tissue. And then a third one, adipose connective tissue. Dense connective tissues have a very tight, dense weave of collagen fibers. If you had to think of them as like a fabric, again, silk, or a really high thread count cotton. So dense connective tissues, they're so tightly woven together, there's not a whole lot of room for blood vessels to supply sugar and oxygen and nutrients. So when they're damaged, they're very slow to heal, if they heal. Dense connective tissue types include dense regular, dense irregular, and elastic connective tissue. Let's start with the loose types first, starting with areolar, then reticular, then adipose. So areolar tissue, this is just a diagram of it, a schematic view of what you might see on a microscope slide, a good one that shows all the stuff and it's properly strained. You got these big and little pink staining collagen fibers running every which way. And then you got these tinier dark staining elastin fibers made out of a protein called elastin that's rubbery. You stretch it out, you let it go, and it snaps back to shape, just like a rubber band. And then there's a lot of empty space filled with water. What do we call the fluid that fills this empty space in tissue? Lymphatic fluid, interstitial fluid, or tissue fluid. It's a different fluid compartment of the human body, separate from blood, separate from cerebrospinal fluid, or anything like that. Okay, then we'll find a variety of cells in here. Most commonly, we'll see these small dark staining things called fibroblasts. Fibroblasts build collagen, they create it. Mast cells and monocytes, these function roughly in the body's defense system, immunity. Technically, in the strictest sense, you cannot find a monocyte outside the bloodstream. Once a monocyte cell exits the bloodstream, it becomes what we call a macrophage. So a hematology specialist would look at this diagram and they'd cringe. There are so many expensive textbooks out there with outdated and by modern contemporary standards incorrect info. It's ridiculous. And when you look at information on the internet, even with Wikipedia, there's so much stuff that's improperly labeled, incorrectly diagrammed, incorrectly stated out there. It's to the point where I used to say, oh, you don't need to buy a textbook if you take my class, you can use the internet as a source. But yeah, I stopped doing that because the internet sources are so corrupt, so wrong on so many things. It's not reliable anymore. So that hammers us hard with monocytes and mast cells. I'll get to that shortly. Okay, so this image is one of our slides in SOS 103. It shows everything that was labeled on the previous diagram. These dark, skinny, purple, black elastic fibers, the bigger, thicker, lighter pink collagen fibers. We have these really dark, compact, angular, dark purple staining fibroblasts here. And then this larger, egg-shaped mast cell. And then this big thing here, which technically cannot be a monocyte, even though it's labeled as such, it's gotta be a macrophage. Yet they're called monocytes here, so whatever. Okay, this image was actually more pink and purple, but I had to zoom in at such a high power magnification that we had what's called chromatic aberration, where the glass in an ultra high power microscope, ultra high powered rifle scope, spotting scope, telescope will bend the light so much that the colors distort. So there's a bit of chromatic aberration here. Extreme far telephoto lenses do the same thing. Okay, so that's a real image of a real slide. Now, like I stated before, these cells labeled monocytes are definitely not monocytes. They're macrophages, which is what a monocyte becomes once it exits the blood and enters the interstitial fluid compartment, interstitial space of the body. So macrophages are like the amoeba. that you saw on yesterday's eukaryotic cell structure module. They're predatory. They phagocytize, engulf, and digest foreign particulate like bacteria, parasite eggs, COVID-19 virions, particles, you name it. There are some textbooks out there that will call these thingies that I call monocytes. They'll say these are the mast cells, and these little granules you see in here are histamines, granules. Others say no, these big clear egg-shaped cells like you see me circling with the cursor here. Others say no, 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 those are the mast cells. So you got two different views here. Mast cells, what they do is they mediate the allergic or inflammation response. They release histamines, and histamines cause vasodilation and more white blood cells to show up at the site of inflammation. So if you have allergies and they're environmental or food allergies and they act up, blame your mast cells for getting a little too hot and bothered. You know, mast cells can be like a small yappy Chiyorki that just yaps when someone 5 houses down on the street sneezes. And, you know, if they're really excitable, you have allergy problems. If your mast cells aren't as excitable, aren't as bothered, you probably don't have allergies real bad yet. Okay, so these other lighter purple staining cells that aren't sharp and angular, that aren't really dark, some textbooks say, oh no, those are fibroblasts. But fibroblasts did make this, and I'm gonna circle 3 of them, highlight 3 of them with a cursor. Number 1, number 2, number 3, number 4. Those really dark staining small cells laid down the elastin fibers and the big collagen fibers. They actually built this tissue by synthesizing the proteins on the rough endoplasmic reticulum, sending it to the Golgi apparatus for modification, secreting it outside their own cellular boundaries. Okay, if we were dissecting real life dead animals, specifically cats, we would see this from a macroscopic non-magnified view, and it has the appearance of cotton or cotton candy, not cotton balls, but just like this layer of fibrous cotton. You find it in between skin and muscle, in between muscle and muscle, when we dissect biological specimens in the lab. Thankfully, because of the lockdowns, I don't have to get my hands dirty with dead animals. Yay! I don't like dealing with the mess and the smell, and I've built up an allergic sensitivity to the chemical preservatives anyhow. Okay, next up is reticular connective tissue. So this tissue contains reticular fibers, which are chemically a different protein mixture than collagen fibers. Reticular fibers stain black when we apply a chemical called silver nitrate. Silver nitrate ain't cheap. A quantity of this much silver nitrate probably run the school $1,500 to $3,000 to purchase. I scored a tiny little amount, and even then it cost me like $30. But silver nitrate is very important in diagnosing the presence of both elastin protein and reticular fibers. So we find this stuff in the spleen and lymph nodes. That's where various types of white blood cells or leukocytes, lymphocytes are made. Oftentimes these reticular fibers in this reticular connective tissue, it forms this curve, these little curving formations like this. So you see these little short arcs and curves and sweeps. And ideally, without silver nitrate, you know, they're going to stain a darker red. With silver nitrate, straight up black. And you look in here, you'll see the place is kind of crowded with cells, lots of cells in here, not lined up in neat rows as if it was epithelial cells. There's no necessarily a large discrete lumen nearby. Oftentimes, this is a very crowded space here. So this is typical lymphatic tissue, lymph nodes. A lot of cells here everywhere, white blood cells in various stages of development. And you see these dark staining silver nitrate staining reticular fibers that sort of form these little spaces, compartments in the background. This is one image. Here's another image. This is just chock full of WBCs. So ordinary connective tissue is not going to be chock full of cells. The cells are few and far between, like with this, like with loose connective tissue. But again, these are all white blood cells. This is where they're birthed. This is where they're made. Okay, if we did not have silver nitrate here, you'd have to look for the same types of shape structure, only with a darker burgundy red staining color. You'd see that there's a lot of circular cells in the background that are all kind of respectably sized in order to determine that, this is reticular CT without the correct silver nitrate stain. So to a degree, this is like art history. Histology is like art appreciation class, art history class, where you look at the color, the shape, the brush strokes, the context surrounding everything, and then you can narrow it down to a time period, a school, and a particular artist, and maybe a particular period in that artist's life. There's a bit of interpretation in histology, especially when the lab texts, the histology texts, don't do their job and correctly apply the correct amount of stains in the correct sequence, which happens a lot with commercially prepared, purchased slides. This is adipose tissue, also known as fat tissue. This tissue type occurs in a lot of places under the skin in a layer called the hypodermis or subcutaneous or just sub-Q layer. It occurs around the kidneys in renal adipose capsules. We find it inside bones where it forms yellow bone marrow in some bones. So even if you're super skinny, have very low body mass index, you're zero fat, you're still going to have adipose tissue inside your bones and around your kidneys. So these are the cells, these big, thin purple lines surrounding this empty white space inside. These are adipose sites, individual cells. And the cell nuclei, let's see if I can zoom in here. There we go. Just dark little blue area here, that dark thing, that dark structure, that dark structure, that dark spot. Those are the cell nuclei. If I had made this slide from fresh material and didn't use any stains or preservatives, you'd see a lime, very light lime green, yellow green material in here. That would be what fat looks like under a microscope. So there would be a fat droplet inside here stored by this individual cell, which is called an adipose site. Okay, the cell nucleus, this little wedge-shaped purple thing, that little dark burgundy colored line, that little purple wedge, that little purple blue wedge, that little blue line. The cell nuclei and organelles are just crowded out by the presence of that huge fat droplet there. Okay, so now for the dense stuff. So as I mentioned before, dense connective tissues have a dense weave of collagen fibers. Loose connective tissues have a loose weave. The difference between silk or high thread count cotton and something like a knit sweater or wool for most wool garments. Okay, so collagen is the most important protein in this particular image. There are other proteins. This is dense, regular connective tissue. All the collagen fibers of the extracellular matrix are pretty much arranged in a parallel fashion. No blood vessels in here, so if this gets injured, chances are good it won't heal. Never mind even healing slowly, it may not heal. So dense regular, you find it forming tendons and ligaments. Tendons join muscle to bone or bone to muscle. Ligaments join bone to bone or muscle to muscle. And if you break a bone in an accident, it'll heal within 120 days. That's 4 months. If you tear a ligament, it might take a year to heal. Even if you go get it taken care of in a hospital with surgery, it's still going to take a very long time to heal after that. And of course, if you tear a muscle or a nerve fiber, that muscle, that nerve is useless after that. Chances are you'll lose that function permanently. Anyway, the fibroblasts, the cells that secrete this tissue, they're so tightly squished in between the collagen fibers they laid down and secreted. They have these, they're just shaped like these little pinkish or purplish lines. They're just squished to these little lines there. So there's a cell nucleus, there's a cell, there's a cell, there's a fibroblast there. Everything else in this image is extracellular matrix collagen. Okay, now this is dense, irregular connective tissue. Yeah, the collagen fibers are densely packed, but they're not all going in the same direction. They're kind of irregularly arranged. And there's other stuff in here. There's enough space for blood vessels and nerves and other things. Dense irregular is really common. We see it on a lot of slides. It forms a reinforcing layer, not just in the stomach, but the entire GI tract called the submucosa. It surrounds bone and forming a layer of periosteum. We find that just below the waterproofing layer in the skin, where this dense irregular connective sheet is called the dermis. So what you see here, this pinkish material, these are all collagen fibers going every which way. Now this circular structure here, this thick wall donut, almost looks like it wants to be a bagel. This is a cross-sectional view of a nerve. Hadn't expected to see this here, but here it is. A nerve shows up. Elastic connective tissue has more than just a collagen. It has the protein elastin. Now, if we're going to look at elastic connective tissue, it would be really nice if the lab tech would stain it with silver nitrate so we could see the dark elastic protein fibers showing up here. But oftentimes you don't get that. This tissue type is found in the walls of large arteries where it has to stretch out and allow the artery to expand when a pulse of blood is pushed through it from the heart. It's also going to form specific ligaments and tendons, such as the nuchal ligament on the back of our neck, and the calcaneal tendon, which runs from the calf muscles of the leg, the triceps surae, gastrocnemius, and soleus, to the heel bone, the calcaneal bone of the heel. Good runners will utilize this calcaneal tendon since it's elastic like a bungee cord. It stores energy, and a good runner, their heel should not touch the ground. Balls of the feet touch the ground, toes, and then the heel lifts off the ground with elastic recoil from the Achilles tendon, giving the body kind of like a spring-loaded assist for running and jogging. I can always tell the noob runners and the runners using poor technique because I hear pound, 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 pound when they're running up and down our neighborhood doing their running or jogging routine. And then I know the pros where they run silently and it's like they're bouncing and just fluttering, very graceful technique. You don't hear their heel pounding the pavement. But the elastic energy stored in this springy material is what powers that technique, that good technique. Now, this was a spec-freaking-tacular slide. Again, I think it was labeled plantar epidermis in our room. I got this image a long time ago, but this is a blood vessel, a decent-sized little artery. It's stained black with silver nitrate. We have an inner elastic connective tissue band, an outer elastic connective tissue band, smooth muscle, dried red blood cells in the lumen, and other connective tissue out here, and then adipose tissue here. There are so many different tissue types in this image, it's insane. If we do not have silver nitrate applied, which, given the way quality of everything in our world is declining, even as features and technology advances, you may see something like this. The elastic connective tissue is so thick, it has its unmistakable zigzaggy shape here, and it's a dark, deep, dense burgundy red color. So sometimes we can see it without silver nitrate. Okay, so here we go. Dense, irregular connective tissue down here, predominating on the bottom half. Adipose tissue predominating on the top half of this slide. And this particular slide was labeled epidermis, skin. Okay, so that's pretty much it for the connective tissue proper. Now, let's deal with bone and supporting tissue. I talk a lot more about bone after our first test when we talk about the skeletal system. So bone tissue is considered a type of connective tissue. The extracellular matrix does have collagen. There are collagen fibers in bone tissue, but it's impregnated with what's called ground substance, a mineralized stone-like rock-hard material called calcium hydroxyphosphate or calcium hydroxyapatite. So the cells that maintain this stuff and take care of it are called osteocytes, and they live in little hollow spaces called lacunae. So anytime you see osteo in a word, it's going to refer to bone. So like I said, we'll cover bone tissue after the integumentary system. So bone tissue, there might be a few little items on questions on bone, like this stuff, on your first lecture exam. But for the most part, that the bone is for the second lecture exam. Now, supporting tissues. There's this thing called cartilage. 3 different types of cartilage make up the supporting tissues. And cartilage is characterized by collagen fibers so small and so fine and so dense, they look like the gemstone opal or moonstone, or maybe a frosted glass appearance on like a frosted glass lampshade or light fixture or something of that nature. Okay, so the cells that lay down that matrix and take care of it are called chondrocytes. And they're also found in these spaces called lacunae. Anytime you see the word chondro, like costochondral joint, chondritis, chondroitin, and glucosamine, you know that, they're talking about cartilage there. Cartilage is another thing you do not want to damage in life because the collagen matrix is so dense and so fine, there's no room for blood vessels to perfuse it and vascularize it, bring it nutrients and oxygen, or bring the cells nutrients and oxygen. So if it tears, bye-bye capability unless your insurance covers the surgery. When I was a little kid, I was in like just out of high school and college, I was still living at home, and I busted my right knee in mixed martial arts. And, you know, they just did the arthroscopic surgery. I wasn't going to let them drop $10,000 or $20,000 because my mom and dad didn't have that kind of money. So to this day, many decades later, I really can't run or jog like I want to. I can't really do mixed martial arts like I want to because one of my knees is bone on bone. That's okay. Cycling, swimming, low impact stuff for the win. Okay, so this is the first type of cartilage. It's called hyaline cartilage. So you look at this photo, which I took, and you look how smooth. Did I take this? I've got so many images in my library. Honestly, I don't remember which ones I took and which ones I didn't. I think I did take this one. You look at this frosted, blurred area down here. It's like frosted glass, or like certain gemstones like moonstone, labradorite, opal. Without necessarily having the fire in the opal, like a good Australian or Ethiopian opal, or Virgin Valley, Nevada opal. I have opal in a oil drilling core sample from Louisiana, believe it or not. Actually, it's nice looking stuff. So this hyaline cartilage, anytime you see cartilage, it's going to be surrounded by a layer of dense irregular connective tissue, and that's going to be called the perichondrium. And then on the other side of that perichondrium, you'll have other tissue types. This white space here is not a lumen. It's just where the thing shrunk as it dried up on the microscope glass while they were making the tissue. When you make a slide, when you make a prepared slide of a tissue, you slice flesh, meat, bone, whatever, so thin that it's going to literally start drying up and spoiling and getting ruined the second you do it. So you got to do it in a humid environment where it's moist. Put it on that glass slide, put water or fluid on there, keep it from drying out like beef jerky. Otherwise, it turns into cartilage jerky, and you get this big empty space here, which is an artifact of the preparation of this histological slide here. So where do you find a hyaline cartilage? Articular surfaces of bones where one bone meshes with another bone. We find it forming the cartilage plates of the nose, the ear, and the larynx, the voice box. Also, it holds open the air passages of the windpipe, trachea, and some in the lungs. Now, fibrocartilage. You know what? Let me do something here. Let's look at this in greater detail. I'm going to zoom the heck in. I know this is my image I took because it's so high res, I can just keep blowing it up and up and up. So these little purple dots, these are the chondrocytes that built what you're looking at. And then you see this space surrounding it. This little alcove is called a lacuna, lacunae plural. And then you can see this frosted, blurry, hazy, opalescent cartilage matrix. So a lot of chondrocytes living in lots of lacunae in here. Okay, let me just shrink this back down or we are going to have problems. And of course, on the edge, you can see these collagen fibers here that have frayed off from the edge of this mass of cartilage, this perichondrium. So let's shrink this down. Otherwise, our PowerPoint is going to not function very well. And go to the next one, fibrocartilage. So again, you see the chondrocytes inside these clear oval egg-shaped lacunae. But what you notice about the matrix in between them is it's not so smooth and frosty. You see these coarse fibers in here. You can make out the collagen threads that stain pinkish red. This was a very nice image I just pulled off the internet because it was just so perfect. And then I went and took some that are nearly this good, close to this good. And I have to figure out where on my slide library they are, where in my stick drive they are, because I didn't label them correctly. Anyway, fibrocartilage is found on the knee joint where it forms cushioning pads called menisci. Also, in between each vertebrae, each bony element of our backbone, vertebral column, or spine, it forms what's called a disc, an intervertebral disc. And then we have some others in other parts of the body, like the pubic symphysis, front where the right and left hip bones meet together. Okay, elastic cartilage. We took silver nitrate, applied it, and it shows up in this middle layer of elastic cartilage here. We have adipose tissue. We have dense, irregular and dense regular connective tissue here. And I'm going to zoom in here. Look at these cells. Look at these red snake, look like red worms. These are smooth muscle fibers making up smooth muscle tissue. So this stuff looks very similar to this stuff up here. I'm circling with the cursor. This stuff up here and right here, this is dense regular and dense irregular connective tissue. But these cells, look how thick they are with a nucleus in the middle. These are smooth muscle fibers. And this looks here to me like smooth muscle tissue as well. So this slide actually has a lot on it. But going to the cartilage mass down here, you can see these threads and bands and struts and strings of dark black staining elastic cartilage. Long Beach City College, the Carson Street campus by the airport, has a spectacular, excellent histology collection. Whoever was running that department years ago did a really good job procuring slides. So I filled in one summer for somebody there and put my microscope cam to good use. Now, your textbook will say that elastic cartilage is only found on the earlobe, the part you get pierced to hang earrings on, or traditionally did. Yet this slide is labeled epiglottis, which is not the earlobe. That's underneath your tongue above the Adam's apple in the throat. So, doing research outside of the textbook teaching, trying to stay current on everything with anatomy, physiology, all other aspects of biology I have to teach. Apparently, elastic cartilage is found in other areas, epiglottis, some components of the voice box or larynx, as well as eustachian tubes, which go from the ear, middle ear cavity to the back of the throat. So the contemporary info out there may disagree with a lot of textbooks, but I've seen lines of evidence going back to the 1970s that the textbooks that say elastic cartilage is restricted to the earlobe only are not correct. So we'll say epiglottis, eustachian tubes, and earlobe. 3 different places in the body where it's found, at least. Okay, so this is the same slide. So again, like I mentioned previously, there's 1, 2, 3, 4 different tissue types that show up beautifully on this image. And yet this slide was labeled epiglottis. I had no idea I'd find all this other stuff looking this nice on one slide. So if we were meeting in person, this would actually be a real headache for you guys, because you'd look at a slide labeled one thing and you'd see something totally different on it. You'd have to search all over that slide for that tiny little area that has what you're looking for. And a lot of instructors never tell their students that, and they lose 25 to 33.33% of the class after they fail the first lab practical on histology. So there's a lot of helpful life hints and techniques to survive this stuff. Okay, so that's it for the connective stuff. Histology is very strongly visual. That makes it hard, but at the same thing, that makes it nice. Now, I'm going to close this PowerPoint. I'm not going to save anything. And okay, pause screen sharing. Let's share our screen with this file. So a library of images, I would walk through everyone in the lab if we had time. So areolar connective tissue, the dark purple things that are skinny are elastic, and the thicker, very faint apricot, peach, pinky orange things in the background, you can make them out if you look closely. Those are collagen. And then let's see, we have these little tiny dark staining fibroblasts. We have what I'm thinking are larger, maybe mast cells. What else do we have? If we zoom in, this here is supposed to be a monocyte / macrophage. Not sure about that, but that's what the books say, so I'll go along with it. And then we have these dark, it is no nice way to say what I'm about to say. These fibroblasts, look at that. Look at that. If I found those behind the freezer that I have in the garage by the HVAC unit and the water heater, I would think I got a mouse problem. They look like little dark colored mouse turds, little tiny little doodads, mouse turds. That's what these fibroblasts kind of remind me of. And then look at this lighter, thicker, more voluminous, light purple textured thing here. Again, that corresponds with what a lot of books would call a mast cell. But again, other books are now disagreeing, other authorities. And then, of course, this is supposed to be a big phagocytic white blood cell, a macrophage. Okay, here Very nice macrophage slash monocyte, but it cannot be a monocyte. It has to be a macrophage because it's not in the bloodstream. It's out in the tissue fluid here. Look at these light colored things here, very light textured oval to egg shaped mast cells. And then supposedly a few smaller, darker staining fibroblasts in the background. Again, different slide, different image. Okay, reticular connective tissue, you saw this one. Here we go, you saw this one with the dark staining reticular fibers. Liver, I'm sorry, not liver, spleen and lymph nodes specifically for this. Look at all these pink staining cells in the background. Look, this is chock full. This is like the mall. This is like the shopping mall on the day after Thanksgiving. This was like all the GameStop and the other stores when the PS5 came out last Christmas and people were buying them for $600 and flipping them on the internet for like $1,800. Hell, I'd have waited in line 6 hours if I could have made a $1,200 profit. That's like $200 an hour take when you think about it. Okay, so anyway, you can totally see all these round red blood cells. White blood cells, leukocytes, lymphocytes, B cells, T cells, eosinophils, basophils, neutrophils, and stuff in the background, surrounded by these really big dark staining reticular fibers. Okay, more of the same. Adipose tissue, already talked about that. Really easy to figure this one out when you see it. Adipose tissue here, adipose tissue there, dense irregular connective tissue up here, a little bit of simple cuboidal here. This is the skin. Hypodermis or subcutaneous fat right here, and then the dermis or the tough, strong, structurally important part of the skin right here. And these are sweat glands. But we'll talk more about that technically on Monday, but I'll have the lectures recorded and up over the weekend. Another image, adipose tissue, dense, irregular connective tissue. You saw this one, mostly dense, irregular connective tissue with a beautiful nerve, cross-sectional view of a nerve showing up right here. Dense, regular. Now again, look at the cells that built this. The fibroblasts are these little purple lines. They're squished to death. They are crushed flat like grapes on a highway. And all this other stuff is ultra densely fine woven, all in the parallel direction, collagen. Okay, here we go. We got a different image. I took all of these different slides in our classroom. And right there, that smudgy dirt on my microscope glass. So fibroblast, 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 there's another one, another one, another one. So you can see they're all lined up in rows on this particular slide. A lot of textbooks will show that image. I just happened to find one in the room and captured it. Elastic connective tissue with the dark staining with silver nitrate, elastin fibers. Talked about this one already. Outer elastic band, inner elastic band. Here we go. I mentioned that silver nitrate is not always going to be properly applied because it's expensive and corporations are cheap. Companies produce these slides for sale to colleges, high schools, universities, medical schools. And look at this. They didn't apply silver nitrate. They didn't have to, actually, because this isn't supposed to be a slide that has silver nitrate. This was that spectacular plantar epidermis slide. This is a blood vessel that was in someone's foot. But this elastic connective tissue layer is so thick and so unique in its shape, you can totally make out the elastic connective, the elastic protein layer, even if there's no silver nitrate and it doesn't stain black or dark chocolate brown or amber color. So like I said, with histology, it's visual, can't always go by color. Sometimes you have to go by shape and context. There is a heck of a lot to talk about in this image, but we'll get to that later in the semester. Well, look at this. a different view of hyaline that wasn't on the lecture PowerPoint. Look at that glassy, smooth matrix, lacunae with chondrocytes living in them. Look at this stuff down here with all these little dark purple lines. Perichondrium, dense, irregular, dense, regular connective tissue that surrounds the body of cartilage here. More hyaline cartilage. There we go. Crazy up close and personal view at very high power magnification. Lacunae, these little egg-shaped white spaces, and we see these little dark staining, darker colored cells living inside there, producing all this collagen matrix to make this hyaline cartilage. And once again, that image I talked about more from Long Beach City College's collection, elastic cartilage with with the elastin staining black with silver nitrate, adipose tissue, all that cool stuff. And a different image, ooh, a thick walled blood vessel, a little bit of elastic fibers in this artery as well. Look at all this dense, regular, dense, irregular stuff out here. So there's a lot of stuff on this slide. Last is bone tissue. So we cover more about this after this first bit of material, but all the textbooks and all the lab manuals show this material with histology. So this is a cross-section of dense bone like you'd find in a thigh bone, femur, lower leg bone, tibia, arm bone, humerus, radius, ulna. And you see these little dark circles. These are called central canals. There's the blood vessels in there. And then you got these concentric rings, these layers of material are radiating away from them. Well, that's all bone ground substance matrix, extracellular matrix, calcium, hydroxyphosphate, and collagen. Now, these little dark lines, let's zoom in. Let's see, can I go to, okay. Well, I'm just going to use this stock photo, expand it a bit. So this dark line here, I'm circling with the cursor, that is a lacuna. Here's another lacuna, another lacuna. Osteocytes are the cells that live inside these, take care of this bone tissue. Okay, so I'm going to shrink this image back down and go to our last one. So this is a bone tissue slide from our room. And again, you can see the lacunae where the osteocytes would live, the central canals, whole bunch of other stuff to say about bone tissue here, but this is not the bone section of the course, so we will leave this till after the first test. Okay, so that was just a little bit of images to view here. I'm going to stop the video now to save bandwidth and space. So stop that recording.